Thanks, Allison. I'm so happy to be here, and thank you for inviting me. So I want to talk to you a bit about how we're trying to harness biological intelligence, so to help us build living machines that have nervous systems. If you look at the animals that currently exist in the world, and that's just a small part of the total space of possibilities, and their nervous systems of these animals. We know that these animals have evolved in the course of evolution and based on natural selection, and their nervous system and the structure and function of the nervous system that they have is really constrained by their body plan and their behavioral niche that they have evolved in. The question that we wanted to ask is that if we take cells from a given organism and put it in a completely new context or embodiment, how do these cells behave in this new context, and to what extent can embryonic cells adapt to a totally new condition and adapt in the developmental time scale as opposed to evolutionary time scale? And can we use principles that we learn of cellular self-organization to build intelligent living machines? And to answer these questions, I've been using frog embryonic cells, and the reason for that, okay, so here's a video showing 1 day of life of a frog embryo. Let's sped up. And so these are really cool because there's a lot that is known about their developmental biology and which cells from which part of the embryo will become what in future, so we can have access to that. For example, it's known that the cells at a certain developmental stage, if you take cells from the top part of the embryo and look at them, those are the ones that are going to become the skin of these little tadpoles in a few days. So, yeah. Another thing that was known before is that if you take an embryo and take those cells, just cut them off of the embryo and put them in a dish in salty water, which is a completely new kind of environment and configuration, what they do is that what biology does really well is that they heal themselves after about an hour or so. It forms like a little ball, and then after a few days, they actually start moving around in a dish like little Roombas. And the reason why they can do that is that they're actually little skin organoids that have multiple cell types, including these multiciliated cells that have cilia that spontaneously beat, and those are little motors that help them move around. So we wanted to ask this question: can we give these little skin organoids brains, and how would they behave if we implant these with little neural precursor cells? Would these cells actually differentiate into neurons? Would they make connections with this motile body? What kind of behaviors will we get? And exactly we did that. We implanted, here you can see little clumps of neural precursor cells inside of this explant. And sure enough, these cells were very sticky. They just remained in there and the thing heals itself. And after a few days, also it starts moving around the dish. So here's a video of one of my favorite neurobots, that's what I call them, that they could make really interesting movement patterns, and I obsessed for a very long time just looking at their movements and trying to see what kind of behaviors they do, how would they be different from the ones that don't have neurons. And here are a few examples. The one on the right is the one I showed you the video on, and then there are different kinds of I call personalities that you can get out of these depending on how you build them, which is currently just me building them manually, randomly getting these behaviors, but potentially these could be used to design various types of movements patterns. And it turns out that the neural ones tend to, in general, move a lot more than the non-neural ones, and their movement trajectories tend to be more complex. If you look on the inside and see what happens to those little circular cells that I implanted, you see on the left, in red, the processes of neurons. They have axons and dendrites that have grown, and they also extend towards the surface of the neurobot. Red is also staining cilia of the multiciliated cells, and the blue just shows the nuclear stain. The question is, do these make synapses? Green is showing a presynaptic marker, and you see that it's also present all over the place. So these are making all these machinery that is you need for neural processing in the nervous system. You can build these using neural precursors that express calcium indicators, fluorescent calcium indicators. On the left, you see that these cells are actually spontaneously active, so you can see the firing. And also, if you treat neurobots with some neuroactive drug, like PTZ, that usually induces seizures in animals, it also changes the behavior and the complexity of movement. So based on all these kind of initial experiments that we've done, we think that it is possible that neurons can control the movement of the neurobot. We also have looked at gene expression pattern, and neurobots show a big upregulation of a lot of genes. As expected, a lot of them have to do with nervous system development, but also we have found that there are upregulation of sensory processing genes, specifically those involved in visual, chemosensory, mechanosensory heat. So we're very, very excited about looking at the behavior of these and how they respond, for example, to sensory stimulation. What kind of network architectures have formed? What are the neural mechanisms that might cause these responses? Can they exhibit fundamental forms of learning, like habituation? And can we train them in the course of development to perform specific sensory motor behaviors? I would like to thank everyone here, my colleagues at the Wyss Institute and the Levin Lab, and thank you for listening.
Questions, comments? Yep. Patrick, I'm coming to you.
Thank you. Fascinating talk. I'm curious, when you mentioned that there are these 4 different behavior types, are those spawned by different methods of creating the neurobot, or are those the same method leading to these different outcomes somewhat randomly?
Yeah, the second. So there are not only 4, there's a lot more than 4. And at this moment, I'm building all of them manually, so and then I see later what happens. But I think these are all, we're in the process of finding what is different in all of these, so in future we can actually say, I want to build one that does pyrographs and be able to build that.
Thank you.
Yeah.
Do we have time for another question, comment? Okay.
Yes, thank you for your excellent talk. A question about perhaps temporality and development is that once you have developed a neurobot, is there further development that takes place, and how does that development take place, both autonomously and through intervention?
That's a really good question. So the way the system works right now, which is actually really cool, makes it really robust, is that embryonic cells of frog have these yolk contents that they can feed on, and that lasts about 10 days. So after 10 days of development, they slowly are going to die. And I'm actually excited to talk about longevity folks about this, because I think this is a great model for those studies. But yeah, so in terms of how far they develop, if you feed them afterwards, they have been kept alive for months. You have to give them nutrients just like other cell cultures. But before that, you don't need that, and the system doesn't even have to be sterile. It's super robust, so it's very cool.